So my talk, which covers the objectives, that is including introduction, microbial pathogenicity, and about the host and parasite interaction, and period of disease, and various infectious agent and their portal of entry, as well as the brief discussion about the bacterial virulence factor, antibiotics mode of action, particularly bacteria, and the mechanism of drug resistance, and also drug sensitivity test, and the conclusion. So when we see the microorganisms, actually they have evolved and they developed a wide variety of tools themselves, and they established the tools themselves to enter into the host to gain the nutrients for their growth and survival. So in this process, the consequence of which results in the tissue damage and eventually disease production in the host. So in order to cause disease, a pathogen must successfully cross the different stages of the pathogenesis. So actually the pathogenesis is nothing but the stages of the different stages of the pathogenesis. So these stages, example via exposure or addition or invasion and infection. So these are the pathogenesis stages. So which will the outcome of an infection is depends on the virulence factors of a microorganisms, that is a pathogen, and at the same time it depends, the infection is depends on the host resistance or the host immunity system. Okay, so the infection severity also varies person to person based on their tolerance, resistant tolerance. So actually, our topic, that is microbial pathogenesis and drug resistance. So these drug resistance present a global health issue now. Okay, so these antimicrobial resistant AMR is a dynamic phenomenon that occurs when microorganisms, particularly pathogen or parasites, they resist the effects of medications when we use the various types of antibiotics and the drugs. So they resist the effects of medications, making the common infections to harder to treat and increasing the risk of the disease spread. So this is a global health issue. Pathogenicity of the microorganisms. So actually the microbial pathogenicity is nothing but a pathogenic microorganism is usually defined as a biological agent. So this microbial pathogenicity has been defined as the structural and biochemical mechanisms whereby the microorganisms cause the disease. So the pathogenic microorganism is usually defined as a biological agent that can cause or damage to its host during the host and microbial interactions. So what is the pathogen means? Pathogen means the microorganism which have the capability to produce a disease. It is referred as a pathogen. It may be includes bacteria, it may be include fungi or a protozoan or a parasite, other helminthes, whatever may be, viruses. Okay, so I have given you the example of the pathogenicity in bacteria. When we see the pathogenicity in bacteria, it is associates with the unique structural components. Their own cellular components is acts for the pathogens to cause a disease to human host or other animal host. So the microbiologist can also be defines the infections or diseases through that parasitic relationship. So every day in our environment as a human, we bombard and we exposed the numerous variety of the microorganisms in the environment. So they may be beneficial, they may be harmful. Okay, both types of microorganisms we have in the environment. Some are very friendly to us, and they are effects in a harmonious way. Some are harmful, which cause the diseases and affects the harmful effects onto the host. So based on the relationships, that is host and parasite relationships. So what is actually host means? Host means when a organism that supports the survival and growth of another organism. Okay, so it is referred as a host. So when we see the interaction of these both is called as symbiotic. The interaction of these both, one is host, another one is depending organism, that is parasite. The interaction of these together, that is called as symbiosis. So this symbiosis refers to the living together. It means living together of the organism. So which includes various types of symbiosis we seen occurs. So that includes commensalism, mutualism, and parasitism. When we see in the parasitism, so in this relationship, one organism benefits and the other one is adversely affected by that parasite. So here in the parasitism symbiotic relationship, one is adversely affecting, but one is benefiting. So when we see in the commensalism, so commensalism, there is the organism, the organism, they cannot harm to the host. So example, you can take our normal flora. So in our anatomical locations, the various microbial flora is present, so they can help us in a harmonious way, and they cannot cause any diseases to us, but sometimes they act as a opportunistic type of pathogens. But actually they are the commensals. Commensals is nothing but it is a normal flora which is present in the host, present in the human anatomical locations. They helps that human to degrade, digest their digest their food material, and also they helps, the normal flora helps to gain a immunity, and they helps in a harmonious way to that host. So that is called as a commensalism. So when we see in the mutualism type of symbiotic relationship, in that mutualism, the both organism will benefited together. Okay, so technically, when we see that parasites are those organisms where they live based on their technically they defined as when that organism that live on or within a host of organisms, they are metabolically dependent on the host. Okay, so if the organisms leaves surface of the host or on the host, that is called as the ectoparasitic. Okay, so if it is lives intracellularly or inside the host, that is referred as a endoparasitic. So that the host on or in which the parasitic organism either attains, which organisms completes their reproduction in a host, which is multiplies a double multiplication in a host, that host is considered as a final host. So where some hosts act as a intermediate host, means they just helps that serves as a temporary but essential environment for some stages of the development of a parasite. So that is considered as a intermediate host, where the parasite is completes the life cycle, where the parasitic multiplies and doubles their number, that host is considered as a permanent host. Okay, final host. So when we see the infectious disease, how we can evaluate the infectious disease? Means when the body, the normal functions are not occurred. Okay, so the some abnormalities occurred in the host, so they are not capable to carry function the normal functionings as usual. Some due to the presence of the parasitic material, due to the presence of their virulence. So actually, virulence is nothing but, what is the virulence? Virulence are nothing but the, it is a quantitative measure of the pathogens. So infection is nothing but number of organisms, pathogenic organisms, and their virulence factors by host resistance. So it is, this virulence is nothing but a quantitative measurement of a pathogenicity, is measured by the number of organisms which is required to cause a disease. So when we see their mathematical expression of infection, so 50% of the lethal dose is the number of organisms needed to kill the off of the host, or the infectious dose ID 50. It is a infectious dose maybe varies greatly among the pathogenic bacteria. You take examples Shigella and Salmonella. They both cause the disease diarrhea in the human. Sorry, but when we see their infectious dose is varies. They both, Shigella and Salmonella, they both cause the diarrhea by infecting the gastrointestinal tract, but their infectious dose of Shigella is less than 100 organisms, whereas the infectious dose of Salmonella is on the order of 1 lakh organisms. Okay, so the mathematical expression of the infection is varies. So maybe qualitative way, maybe quantitative way. So every day we pampered, we exposed to the various types of microorganisms. So when you take the qualitative way of infection, so one person, if the person is HIV positive person is there, so he will affected even though with a normal flora also. The person who are lowered, who have the lowered immunity, who are the suppressed immunity, example HIV patient, they are suppressed, they have that lowered immunity, that's why they are affected with the normal flora also. So we see in the healthy person, they cannot see, affected more severe, so when comparing to the HIV patient or lower immunity person. So when we see the quantitative type, in the quantitative type, that is based on the quantity of the organism present or quantity of the pathogenic virulence present, it may be described. So when we see the mechanism of the pathogenesis, so mainly the mechanism of the pathogenesis is virulence. Okay, so virulence, they have the individual characteristics which called as a virulence factors. So virulence factors play an important role in the pathogenicity. When we see the bacterial virulence factors, example like capsules, villi, toxins, so some enzymes, some secretions, the cellular secretions, they all involves in the mechanism of the pathogenicity. So if we see their infectious agent portal entry maybe varies, okay, individual species, individual infectious agent, they varies to enter into the host. The portal of entry may varies, like example you take the various routes are there to enter into the host, like oral, respiratory, genitourinary tract, conjunctival, cutaneous. So oral means nothing but when we ingest a contaminated food, or when we take that orally the contaminated water, they also helps to entry of that pathogens into the host. So respiratory, when we inhale the inhalation, due to the inhalation also the numerous various types of microbes inhaled with the air, and they also some of the organisms act as a pathogens. So genitourinary tract, which is a suitable for pH, contained pH is very suitable for the pathogens. There, that is also one of the route to entry into the host. Conjunctival, it is a eye, so conjunctiva. So the mostly the eye infection caused by that various types of pathogenic bacteria. Staph areas also. So cutaneous is nothing but skin. Okay, the surface area, epidermal area, surface membranes of the skin which is affected. Okay, so when the scratchy or injury is occurred or caused on the skin. So there is the easy to entry the bacteria inside of the host. So in the image, you can see the various routes to enter the infectious agents. So it is referred as a portal of entry. So in the so evolution of the infection is in the majority of the instances, the parasites get destroyed by the host defense system. So when the pathogen is enters into the host, invasive the host tissues and damage the host tissues, and after that they can enter into the bloodstream and they targets to reach the other organs through that bloodstreams. So in the majority of cases, the parasites get destroyed by the action of that host defense system. But sometimes that infectious agents may escape from that host defense mechanism. So when the host resistance fails, what happens? An acute inflammatory reaction at the site of invasion occurs, characterized by the vascular dilatation, marked exudation of plasma, and accumulation of that polymorphonuclear leukocytes. So some secretions like pus producing. Okay, so the pus producing bacteria are termed as the pyogenic. So example I given here to you, the Staphylococcus aureus is one of the example of the pyogenic bacteria, and another one is the Streptococcus pyogenes. So these are the some examples which are accumulates with the pus producing. When we see the periods of disease, so after invading that pathogen into the host, so some incubation period. After the incubation period, what happens? The prodromal period is occurred. What is prodromal period means? The prodromal period is occurs after the incubation period of that infected agent. After entering into the host, they may take some incubation period to develop themselves there at the suitable environment. So this prodromal period occurs after the incubation period. During this phase, the pathogen continues to multiply and the host begins to experience general symptoms like illness. So which typically result from activation of the immune system. So such as symptoms like fever, pain, throat soreness, and some swelling, inflammations. Okay, so usually such symptoms are too general to indicate a particular disease. So after the following to this prodromal period, the other period, that is period of illness. Here the severity of the symptoms are occurred. Okay, so during this period, the symptoms of the disease are most severe. So why? Because in the prodromal period, they multiplies more number and they secretes their toxins. So a period of illness is nothing but they completely the host tissue is damaged and the symptoms like the severe symptoms are occurred during this period. So after period of illness, so the period of declined period. So the follows period of decline during which the number of pathogen particles may be decreased due to the action of some immunity and due to the action of some antimicrobial agents when we use some antibiotics. So due to the reasons of various reasons, maybe they became decreased. Okay, so and the symptoms of the illness also begin to the decline. So however, this during this decline period, the patients maybe become susceptible to developing the secondary infections. Why means actually the post that patient is already suffered with the primary infection and lower weakened by the primary infection. That's why that patient may be becoming susceptible to the developing the secondary infection because of their weakened immune system due to the reason of the primary infection. So the final period that is the convalescence period. So during this stage, the patient generally returns to the normal functioning. Okay, so in some cases, also some diseases may placed as a permanent damage and that the body cannot fully repaired. But in some cases, they may be returns to the normal functioning. So this is about the periods of the diseases. So coming to the our main thing, that is the microbial pathogenic factors. That is, I want to discuss about the bacterial virulence factors. Okay, so the capsule, one of the most important and virulence factor that is surrounds that bacterial cell wall. Okay, so this outermost, the slime layer, gelatinous layer, which is produced by the bacterial cell wall actually. So this capsule involves and plays an important role in the virulence as a virulence factor. Okay, so when we see the mechanism of the action of this capsule helps and protects the bacteria from phagocytosis. Understand? Not only it protects from the phagocytosis, it is also helps the bacteria to protect from the outer environmental, unfavorable environmental conditions. Okay, so what is phagocytosis? So phagocytosis, it is nothing but a engulfing of a foreign particle. So it is a mechanism of engulfing a foreign particle. So whatever may be, not only the bacterial cell, when the foreign particles or dust particles or even the other foreign body, which is uh, attached to that macrophages or the other white blood cells. So what happens? The phagocytosis mechanism is occurs. So phagocytosis occurs after the foreign body or bacterial cell attached to that cells. Okay. So what in this process, the phagocyte, they stretches itself around that bacterium or around that the foreign body particle and engulfs it then. Okay, so these phagocytosis is a critical part of the immune system. So several types of the cells of immune system, they involves, okay, performs such phagocytosis. Example, you take neutrophils, macrophages, dendritic cells, and B lymphocytes. They all performs such type of the phagocytosis. So during these phagocytosis, that phagocyte stretches together and surround the around itself stretches and around the bacterium, engulfs the bacterium and releases some of the lysosome enzymes or bacteriocins. So by the effect of the enzymes and secretions, maybe that foreign particle or the bacterial cells degraded or damaged or killed by that phagocytosis, actually the during the mechanism of the phagocytosis. So in case of that capsule containing bacteria, they escape. Here you can see in this image. So capsule helps to prevent interaction between the antibody and the C3 bound, which is present on the outer membrane of the bacteria. So actually the cell wall contains the C3b receptors, okay, that sites, binding sites, C3b binding sites, which is actually binds to the receptor of that phagocytic white blood cell, this orange colored one, receptor C3b. So in the normal functioning, these C3b receptor and the capsule bounding site may be, the both are come together and bound together and undergoes the phagocytosis performance or mechanism. But in case of this capsule containing bacteria, this capsule prevents the interaction between the antibody and that C3 bound to the outer membrane of bacteria and their respective receptors on the phagocytic cells. So what happens? They escapes and they prevents to that phagocytosis. That's why they easily escape that phagocytosis mechanism and they ready to cause effect to the cells of the host. So here the capsid, the slime layer, biofilm, they are helping and protecting the bacteria from the interaction between these antibodies. So like that, the capsule, one of the virulence factor helping the bacteria, protecting the bacteria from the interaction between the phagocytosis action. The another factor that is pili, okay, in the adhesion, okay. So adhesion is nothing but the adherence to the surface particle, surface area of the host. So these adhesins, the pathogenesis of many microorganisms, they adhere to that mucosal membranes or mucosal cells of the host. So when we see in the intestinal tract or in the bladder or blood vessels, so that these factors like villi and other adherence factors of the bacteria, they adheres to the surface epithelial cell linings in the evenly in the intestinal tract walls, epithelial walls, and that blood vessel walls. So which would have been otherwise washed away before they could cause disease. But by the reason of their factors, okay, adhesion factors, they are not washed out, okay, but which would have been otherwise washed away before they could cause a disease. So these factors, adhesion factors, helping them to adhere the surface of that host tissues and host cell linings. Example of adhesins, types of adhesins, one is pili. So in most cases, the pili serve as an adhesion factor. So many of the adherent proteins are present at the tip of that pili. Pili also referred called as a fimbriae. So and they bind tightly to the target tissue. Okay, so by that reason, they can easily damage the host layers, host epithelial layers, and they enters into the host cell. Okay, so here you can see in this image, okay, the fimbriae, how they are present the outer side of that cell wall. Okay, so the structural adhesions of gram-negative bacteria are known as the fimbria, and those of gram-positive bacteria are called as the fimbrillae. Okay, so not only pili, so the various other types of factors which is helping the bacteria to responsible for the pathogenicity. So like example, LPS, lipopolysaccharides. So these lipopolysaccharides are nothing but it is the outermost layer, additional extra outermost layer of the gram-negative bacteria. So where uh, when we see in the gram-positive bacteria, there is no the outer layer membrane like this. Okay, so there is the in the gram-positive bacteria, there is only the polypeptides. Okay, that peptidoglycan is present the outer cell wall. Okay, but in these gram-negative bacteria, they consisting the outer membrane. Okay, so it is have that some toxin effect. So it is also called as endotoxin. Okay, so it have the endotoxin activity, and they contains the O chains and core protein polysaccharides, O polysaccharides. So they are responsible to in that pathogenicity as a endotoxin. For example, you take the gram-negative bacteria, they contains this outermost layer, which is called as the lipopolysaccharides. They also helping the pathogenicity. Okay, and M protein. M protein is nothing but it is a one of the layer. Okay, M protein is a virulence factor, so it can be produced by certain species of the streptococcal organism, streptococcus. So what happens in the bacteria? They covers the bacteria is covered in the protein M protein and sugar molecules. So these M proteins that help them gain entry into the host by that counteracting the host defenses. So these M protein also helping as a capsule, like a capsule that is also helps from the escaping from that defense system mechanism, and helps to adherence to the epithelial cells. So in this image, you can see it is nothing but a pharyngitis, cause of pharyngitis in that oral cavity. Okay, the pharyngitis is infected with that number of streptococcus pyogenes, streptococcus pyogenes. So invasiveness. Okay? So the invasiveness of bacteria. appears to be multifactorial. Okay, so it is a complex process, not in a only in single way, but it is a complex process. So invasive bacteria either destroy the barriers of the host cell and or penetrate into the cells of the barrier. So you take example that Shigella, a series of invasive proteins are involved here. So these invasion plasmid antigen, this protein is called as invasion plasmid antigen IPA. So which is expressed on outer membrane of uh, bacteria. So permit the bacterium to bind to the luminal surface of the mucosal and cells of the intestine. So this protein helps to the Shigella to enter into a host cell easily. Okay, so not only Shigella, in some cases, the another protein like example that is ICS, intracellular spread protein, which helps to the bacterium. It is expressed to outer membrane of the bacterium and allows to the interact with the host cell deeply, okay, integrins, and following the interaction of the bacterium, which is helps to travels towards the another the adjacent cells. So by means it is by means to through that protrusion. Protrusion is nothing but the hernial. So the one cell membrane which forms a hernial protrusion and fuses with the cell membranes of the another adjacent cells. So thereby what happens? The enabling Shigella to diffuse from one cell to the another adjacent cells. So like that, they spreads the infection from one cell to the another cell. The invasive the from one cell to the another adjacent cell. So when we see some enteropathogenic bacteria like Yersinia, so the invasion protein which promotes the binding of the bacteria to the host cells. So here they stimulates the cell to invaginate and take into the bacteria. So Yersinia bacteria is taken into the host by the help of some stimulates, some proteins. Okay, so that proteins helps the bacteria that at the site of promoting binding site, they allows that bacteria to invasive inside of the host cell. In coming to the Neisseria gonorrhoeae, so with the help of pili or fimbriae, so they adhere to the mucosal cells. So these pili also helps that bacteria, some enzymes that dissolve some mucosal cells lining which is present on that epithelial cell and thereby enables that Neisseria gonorrhoeae to penetrate the submucosal tissues of the host cell and helps them to entry into the host. So not only the cellular components, the many various enzymes also play an important role in the pathogenicity. Okay, so like example, exoenzymes. So many bacteria have the ability to secrete or release enzymes that can damage the host tissues in a variety of mechanisms. So one you see that enzyme that breaks down the collagen. Okay, the example that is collagenases. So collagenases have the ability to break down the collagen. So by that collagen breakdown, they can easily damage the host tissue. Okay, not only collagenases, you take example the hyaluronidases. So actually, what is hyaluronic? Hyaluronic acid is present the connective at the site of connective tissues of joints of the human. Okay, so at that, so the whole body connective tissue contains this hyaluronic acid. So these hyaluronidases acts on that hyaluronic acid and degrades them and allows that pathogen to enter into the host connective tissues and cause the disease. So the second one, enzymes that break down the cellular materials. So the first one, enzymes break down that collagens and hyaluronic acid and that fibrin, the blood clots also be degrades at the tissue. Okay, so coming to the second one, the enzymes that break down the cellular material. The completed cellular material will be degraded by the action of that exoenzymes released by the bacteria. Example, you take the proteases, lecithinases, which are associated with example in the class Clostridia species. They are able to produce such type of enzymes. They can easily break down the cellular materials and they enter into the host tissues and they cause the diseases. So not only the enzymes, the many bacterial pathogens, they have the ability to produce the toxins. So the toxins have the effect of the toxicity. So they may be varies. So some are cytotoxins, some are neurotoxins, some are enterotoxins. So enterotoxins are nothing but which affects the inside of the intestinal tract, gastrointestinal tract. Such type of toxins we called as the enterotoxins. You can take example cholera toxin, E. coli toxin. They all comes under this enterotoxins. Particularly the enteric bacteria, they can affect such type of gastrointestinal tract by the action of the enterotoxins. You take the neurotoxins. Neurotoxins only affects to the nervous system. So example, you take botulinum toxin, tetanus toxin, that affects the nervous system. So cytotoxins, which is released from the cytoplasm of that bacteria, and they affects the variety of the tissues. Okay, so the another one is lipopolysaccharide. Already I explained you that endotoxins, which are present inside that cell wall of the gram-negative bacteria. When the bacteria undergoes the lysis, then they release that endotoxins outside, and they also responsible for various sepsis and septic shock. Sepsis is nothing but the bacterial infection in the blood is called as sepsis. Septic shock is when that organs become that damage to functioning, okay, abnormality in the functioning, that is septic shock. So endotoxin mediated toxicity includes fever, activation of complement thrombocytopenia, and other coagulation perfusion to major organs, shock, and it also leads to the death. So these are some various types of that virulence factors helping in the and contributing for that pathogenicity in the various pathogenous bacteria. So the next thing, that is mode of action of various antibiotics. So we are actually using the various types of antibiotics to reduce the infection or to kill the bacteria or to reduce their growth, to inhibit their growth. We use the various different types of antibiotics in our daily life. So when we see the antibiotics, there are two various types of antibiotics we are actually currently we are using. That is, some antibiotics either destroy the bacteria, so that is called as bactericidal. So bactericidal antibiotics is nothing but that antibiotic is completely destroy or kill the bacteria, that is called as bactericidal antibiotics. So whereas some antibiotics are not kill the bacteria, okay, but they only involves in the slowdown or inhibiting the growth of the bacteria, that is referred as the bacteriostatic. Okay, so the both types of antibiotics currently we are using. So here I have given you that general image of the bacterial cell. Okay, so bacteria, they contains the cell membranes, cell wall, and the cytoplasm. Inside the cytoplasm, they contains the chromosomal DNA and ribosomes, as well as extra chromosomal DNA, that is plasmid. Okay, so they also contain some villi and the outer some slime layers like capsule and flagella. So this is about the one image of that bacterial cell. So when we see the drugs, how they affect the mode of action, how the mode of action the various drugs we will see. Okay, so you take example quinolines. Okay, so these quinolines are nothing but the drugs of nalidixic acid class of drugs. So they inhibit the DNA synthesis. Understand? So the quinolines mostly they inhibit the DNA synthesis. So these quinolines are nothing but the nalidixic acid class of drugs, and these antibacterial actions is interferes of that DNA gyrase activity. So they interfere at the DNA gyrase activity. Actually, what is this DNA gyrase activity? So this gyrase A protein helps to attach to a 5' end of that broken DNA. Understand? So they helps the covalently bonding to the 5' ends. So these quinolines is interferes at that region, and they inhibit the DNA synthesis, and they inhibit the DNA sequencing, supercoiling. So due to that reason, that DNA synthesis is blocked, DNA synthesis is inhibited, the bacterial DNA synthesis by the action of these quinolines. So by this reason, what happens? The bacteria may be slow down their growth. Okay, so by the reason of quinolines, they slow down their DNA synthesis. They inhibited their bacterial DNA synthesis. Due to that reason, they decreased their number and they slow down in their growth, but they are not killed by the quinolines. So such type of drug, what we call them, like bacteriostatic. These quinolines are a bacteriostatic type of antibiotics. The mode of action is bacteriostatic. Just they helps to inhibit the growth of bacteria by the action of that inhibiting the DNA synthesis by interfering in the DNA gyrase activity in the covalent bonding of that 5' ends. Okay, and inhibit the DNA sequencing. So the another type of antibiotic you see here, that is beta-lactam antibiotics. So beta-lactam antibiotics, the best example of beta-lactam antibiotics are cephalosporin and penicillin. So these both antibiotics, cefamycin also one of the beta-lactam antibiotics. So these beta-lactam antibiotics mostly disrupts the cell wall. Okay, so how means these antibiotics act by binding to the penicillin binding protein. Okay, so they binds to the penicillin binding protein, and they this penicillin binding proteins shortcutly we called as PBPS. Okay, PBPS. So after binding to that penicillin binding protein site, and they disrupt that peptidoglycan cross-linking. Understand? So they interferes in the peptidoglycan synthesis, the cell wall synthesis. They interferes in the peptidoglycan synthesis. That means cell wall synthesis. And during these cell wall synthesis, resulting in bacterial lysis and cell death. So how means that cephalosporin is binds to that transpeptidase. They interferes that transpeptidase enzyme action. Understand? So what is this transpeptidase? This transpeptidase, it is an enzyme catalyzed the final cross linking in the peptidoglycan cell synthesis. So by the action of that beta-lactam antibiotics, that transpeptidase are disturbed. Their cross-linking of that peptidoglycan are disturbed by the action of these beta-lactam antibiotics. Due to that reason, that will be disrupted that cell wall, okay, cell wall of the bacteria. So it leads to the osmotic effect, which shrinken the cell wall. Complete cell is shrinkened over to shrinkened and become killed, become dead. Okay, so due to these lactam ring contained antibiotics, they more actively involves in the at the region of that penicillin binding proteins, and they disrupt the peptidoglycan cross-linking synthesis of that peptidoglycan, and they make the bacterial cell to die. Okay, so that's why such type of antibiotics is called as bactericidal antibiotics. Why? Means these antibiotics destroying the bacterial cell, okay, by the action of the antibiotics. Okay, so another type, you take example, another type of antibiotics, example, rifamycin. Okay, rifamycin, how they uh, uh, reduce the that bacterial growth means they prevent the RNA synthesis or protein synthesis. So this rifamycin drug is binds in the polymerase subunit. Understand? So it inhibits the bacterial DNA dependent RNA polymerase. So it completely, the rifamycin prevents that DNA dependent RNA polymerase mechanism due to binding in that polymerase subunit deeply that RNA channel, facilitating direct blocking of that elongating RNA and used to treat, actually this rifamycin is used to treat in that diarrhea disease. Okay, so travelers diarrhea, we use the rifamycin. So if this rifamycin is uh, interferes into that RNA synthesis of the bacterial RNA synthesis by binding to that DNA dependent RNA polymerase subunit. Okay, so some antibiotics mode of action is very interesting mode of action they have. You take example erythromycin, they only act on the 50S ribosome of the prokaryotes. Okay, and the another antibiotic, that is tetracycline. Tetracycline or streptomycin, they act on only the 30S ribosome subunit. So due to these effect, antibiotic effect on that ribosome, what happens? The bacteria still live, but they cannot be able to make their proteins. Okay, so it is also comes under the bacteriostatic type of antibiotics. The another antibiotics may involve in the metabolism of the bacteria. So that is nothing but sulfonamides and trimethoprim. So these sulfonamides and trimethoprim, they both inhibits the PABA pathway of that metabolism of the folic acid. So these two antibiotic drugs act synergistically. Synergistically means they both together acts on a target, same target. Okay, so that is referred as a synergistic action. So the sulfonamides trimethoprim inhibits that PABA means nothing but the para-aminobenzoic acid. Okay, so the para-aminobenzoic acid pathway, PABA pathway in the metabolism, folic acid metabolism of the bacteria. So actually, folic acid is necessary to synthesize a nucleic acids. So for synthesis a NADP, ATP synthesis. So without folic acid, there is no synthesis of the nucleic acids. By means what happens? The DNA synthesis will stop, inhibited. So due to that reasons, the bacteria will maybe live, but they cannot able to synthesize their folic acids, and they may not able to synthesize their nucleic acids. So due to that action, the antibiotics act as a bacteriostatic. Okay. So some antibiotics, you take polymyxin example. So these polymyxins actually disrupt the cell membrane. So these polymyxins, the two types of polymyxins are there, B and E. Polymyxin B and polymyxin E. These polyenes are mostly act on fungicide, as a fungicide, fungal cell. These polymyxins are also called as colistin. Also we called as colistin. Why? Means they are works on the membrane, cell membrane. Okay, so they work mostly by breaking up that bacterial cell membrane, and they involves in the disrupting the cell membrane. After that, what happens? They can damage the cell wall, and due to that disrupting the cell wall, the cell become the ruptured, and due to the osmotic pressure, that shrunken and the cell is become die. Okay, so it is this polymyxin also comes under the bactericidal type of antibiotics. So these are various types of mode of action of the antibiotics I have discussed till now. So actually, what is that antimicrobial resistance happens? Okay, including you take the bacteria, fungi, viruses, or parasites. What is actually happening? Means that bacteria capable to change and are still able to grow even when they are exposed to the antimicrobial medications. Okay, still when they are exposing to the various types of antibiotics, antimicrobial medicines, but still they are working, still they are surviving by means they are changing, altering themselves. Understand? Okay, so that are meant to actually antimicrobial medication is for kill or limit their growth, as now we discussed, such as antibiotics mode of action to reduce their growth, to limit their pathogenic bacterial growth or kill that bacterial cell. Okay, but what happening? These antimicrobial resistance changing, altering their composition or chemical composition of bacteria and making them very resistant to various antibiotics. Okay, not only a bacteria, which bacteria, for bacterial we called it as antibiotics, for fungal we called it as antifungals, for a virus we called it as antivirals, for malaria causing agents we called it as anti-malarials, antimicrobial agents, and for helminthics we called it as anti-helminthics, whatever may be. So what happening day by day, they becoming a resistant to the various antimicrobial agents. Okay, so as a result, what happens? The medicines become ineffective. Understand? By the reason of these changes altering in the pathogens, the medicines also becoming ineffective, and the infections persist in the body and increasing the risk of spread to others easily. And while antimicrobial resistance refers to all microbes that resist to the treatments which is designed to destroy them. Okay, so antibiotic resistance is specifically deals with the bacterial species. Okay, bacteria that are resistant to the antibiotics. Sorry. So actually, what is the drug resistance means? These drug resistance, emergence of drug resistant bacteria, it is really a current major problem in the antimicrobial therapy. Okay, so the frequency of these varies with the based on the species and the antibiotic used. Okay, so the frequency is maybe varies based on that organisms and the various types of antibiotics used. So when we see the drug resistance is two types. Okay, so when we see the individual pathogenic species, drug resistant species, the drug resistance is two types, that is primary drug resistance and another one is acquired drug resistance. Understand? Primary drug resistance and acquired drug resistance. So there are four major mechanisms that mediate that bacterial resistance to the drugs. One is bacteria produce enzymes that inactivate the drugs. The another one is bacteria synthesize modified the targets against which the drug has no effect. The another one is bacteria decrease their permeability. Understand? They decrease the permeability such that an effective intracellular concentration of the drugs, which is achieved to the target. And the another way, the bacteria actively export the drugs, okay, by the various pumps. We called it as multi-drug resistant pump. Okay, so most drug resistance is due to the genetic change in the bacteria, in the bacteria, like either a chromosomal mutation or some acquisition of a plasmid or transposons. Okay, so most drug resistance due to the some mutations and gene transfers. So primary resistance includes that some bacteria which is having that possess an innate property of the resistance to the certain drugs. Innate property means themselves from their birth. Understand? From mother cell, they gain that ability of the resistance, the primary resistance that is carried by the mother to the baby, like the mother cell to daughter cell. Understand? The primary resistance is referred as innate property. So like innate immunity, what we discussed in the immunity, innate immunity is naturally gained from our mother womb. Like that, here also some species, they are acquiring the primary resistance as the form of innate property from their mother cell. Okay, and the resistance to certain drugs. The second one is acquired type of resistance. So acquired type of resistance is results either from mutation or gene transfer. Mutation is due to their replication, they undergo the various types of mutation, they changes their genetic material from one side to other side, or the gene transfer. The spontaneous mutations is relatively less frequent, but the transfer of genes either through chromosome or plasmid. Plasmid plays an important role in this acquired resistance to spread of the drug resistance, not only in one bacterium to bacterium, but these plasmid have the ability to spread the resistance gene to the various other outside species also, outside other genera also. So this is a very unfortunate thing. That's why day by day the resistance of the various bacteria to the against to the antimicrobial agents is increasing day by day by the reason of such type of plasmids which are carrying the resistant genes from one bacterium to another bacterium. Not only one group of bacterium, they also have the ability to spread it to outside the other genera also. So when we talk about that antimicrobial mechanism, innate type, so the penicillin actually acts when we take the penicillin antibiotic, it have the less effective on the gram-negative bacteria when comparing to the gram-positive bacteria, because based on their cell wall composition, based on their composition, cell wall composition. What is the difference in their cell composition means in the gram-negative bacteria, they containing the outer membrane, additionally outermost membrane that is protects from the effectiveness of the penicillin. Where in the gram-positive bacteria, there is no present the outer membrane like gram-negative bacteria. So directly they have the outermost, that is peptidoglycan. So these penicillin more effectively act on that proteins of that polypeptides, peptidoglycan. So they have the more effective on the gram-positive bacteria. They kill the gram-positive bacteria cell, so act as a bactericidal on that gram-positive bacteria. When comparing to the gram-negative bacteria, the gram-positive bacteria, the penicillin effect is more on the gram-positive bacteria by their innate composition of the cell wall, and where we see in the gram-negative bacteria, there is a less effective of the penicillin on the gram-negative bacteria based on their cell wall composition. So the another thing that is innate type, we see the bacterial chromosome is okay. So plasmid, okay. So the plasmid is nothing but the extra chromosomal circular double-stranded DNA molecules. So these plasmid have the ability to replicate independently. So these plasmid have the ability, without cell also, they can have the ability to replicate themselves independently. That's the reason they can spread over to the from bacterium to bacterium and other genera also. So these plasmids contains the R gene, resistant gene. So these plasmids contains that resistant genes and carries that resistant genes to the another bacterium. Understand? So they can be transferred not only to cells of the same species, but also to other species and the genera. So these two categories of these plasmids. One is the large plasmid. So another one is the small plasmid. The two broad size of categories they have. Okay. So one is the large plasmid. So they are involved in the conjugation. Another one is the small plasmid that is only gives the resistant genes. Okay. So here in this image, you can see these plasmids containing the resistant genes. So these plasmid we also called as resistance factors or R factors. They are the promoter to drive target gene expression and the target gene, and they also contains the antibiotic resistant gene. So what happens that plasmids in their mechanism, the transfer of the whole plasmid to the other bacterium. So this means that a bacterium can become resistant to multiple antibiotics when it picks up that single plasmid. So they then become multi-drug resistant. Furthermore, genes that influence bacterial virulence are also frequently found on that plasmids. So that pathogenic bacteria carrying multi-drug resistance plasmids have increased in prevalence and now cause the major problems all around the world. It is a major problem. Okay. So in this acquired resistance, there is the two types of gene transfer is occurred. Okay, one is the vertical transmission of the genes, another one is the horizontal transfer of the genes. Which genes? That resistant genes. So the acquired resistant genes is carried out by the two ways, and one is the vertical transmission, another one is the horizontal transmission. So in this vertical transmission of gene that can occur between mother and her offspring at birth. Understand? So during this period, the spontaneously due to antibiotic exposure, while there have been multiple studies about the horizontal transfer of antibiotic resistant genes, but not many studies have been conducted to study this vertical transmission. So in this vertical transmission, what happens that resistant gene is gained during that replication, okay, from their mother cell, they can get in the offsprings. So this is vertical way of the transmission of genes. Okay, so spontaneously it is occurred due to the replication of the cell, bacterial cell. Where horizontal transfer of genes, it occurs in three ways. Okay, so one is the conjugation. Okay, another one is the transduction, another one is transformation. One is the conjugation, another one is that transduction, another one is the transformation. So these three are comes under the category that is horizontal transfer of the resistant genes. It is a acquired resistant, acquired type of resistant. Okay. So when we see in that conjugation, so in this conjugation, what happening? The pilus, okay, the pilus, it is a pilus which helps some porins, pilus, they attach to the other bacteria. So this pilus helps to transfer the resistant plasmid resistant gene to the another cell, new bacterial cell. So the pilus is attached to the bacterial cell and passed on to that another bacterium. So these pilus conjugation pili pilus act as a sex pilus, and they transfers these that R factor to the another bacterial cell. So this is conjugation, called it as conjugation. Coming to the another one is that transduction. Transduction is particularly occurred by the bacteriophages. So when the viral phage is infects to the one bacteria, what happens, that virus attacks to the bacteria and inject its DNA into the bacteria. So this bacteria we called as a phage DNA. They synthesize their phage DNA. Along with their phage DNA, they also contains their these resistant gene which is present in that infected bacteria. So these phage bacteriophage also carries, along with their genetic material, they also carries the resistant gene. When that phage is go and attack to another new cell of the bacteria, then what happens, they again inject their genetic material inside the new cell of the bacteria along with the resistant gene. Like that, they transferring the bacterial DNA from by the phages from one bacterial cell to another cell. This is one type of another type of the horizontal transfer of the genes in the acquired resistance of the that resistant, acquired resistance. The another way, that is transformation. Okay, so in the transformation, what happens? Example, dead bacteria. If it is a dead bacteria, okay, so when that dead bacteria is contains the R gene, okay, the dead bacteria is contains R gene. So some due to some environmental forces or some mechanical forces or physical environmental forces that may lyse, okay. So due to that lyse, they release the resistant gene. So that resistant gene is picked up by the another new bacterial cell. So directly, the free of transfer, understand, free of transfer of that resistant gene released of the DNA by the donor cell and uptake by the another one is recipient cell, right one. Okay, so this is a mechanism called as transformation. Okay, so these three mechanisms which are helping to transfer of that resistant gene from one bacteria to another bacteria. Okay, so these antimicrobial mechanism, the antimicrobial resistant mechanism, which helps they carries that resistant genes, they helps the bacteria to uh, uh, to alter the changes in their composition or in their targeting genes, okay, targeting sites, etc. Okay, so how means, here we see that stop the antibiotic from reaching its target. So these resistant genes helps the bacteria to stop that antibiotic from reaching the site. 1 minute. Sorry. So these resistant genes helping the bacteria for antibiotic reaching its target. How means, the pump the antibiotic out from that bacterial cell. So bacteria can produce some efflux pumps, understand, in their membrane cell wall, in their cell wall, which will actually, they have that motive forces like protons, which is imported, exports, like that. So such pumps. So here in this pumping that antibiotic material, also bacteria playing an important role to produce that pumps, so that sit in their membrane or cell wall. So these so-called efflux pumps. Why? Because they are effluxing out, and the bacterial cell is importing outside the antibiotic which we are use. Example, you use tetracycline for a particular bacteria to kill bacteria. So that bacteria is resistant to build a efflux pumps, and they have the ability to build a pump to export that tetracycline from that efflux pumps. So are very common in bacteria and can transport a variety of compounds such as signal molecules and nutrients transports uh, through that plasma membrane. Understand? So some of these pumps can also transport antibiotics outside, outside from the bacteria. In this way, lowering the antibiotic concentration inside the bacterial cell. In some cases, the mutations in the bacterial DNA, they can make the bacteria produce more of a certain pump, which is called it as multi-drug resistant pump, MRT. So they are all, due to these reasons, the effect of the antibiotics is lowering. Okay, and the antibiotic is lowering to reach their targets. Okay, and this is one reason. The another thing, decreased permeability. So decreased permeability of the membrane also plays an important role in the bacterial cell resistance to various antibiotics. So such a decreasing permeability changes in the bacterial membrane make it more difficult to pass through the antibiotic. In this way, what happens? The correct concentration of the antibiotic may not reach to that bacterial cell. Due to it, it leads to the decreasing the effect of the antibiotics on that bacteria. In this image also you can see they altering their target alterations, okay, and alternating the enzymes and decreasing the uptake of that antibiotics, and their efflux through that efflux pump they are exporting the antibiotics from the cell wall. Okay, these are some mechanisms by the resistance. So the another thing that is the plasmids, resistant plasmids have the ability to code that carry the genes for a variety of enzymes. Okay, that can degrade antibiotics. Understand, Amma? They can degrade the antibiotics, and they can modify the membrane transport systems also. So these resistant genes for a variety of enzymes, they helps to code the secretion of the enzymes. Like example, you take beta-lactamases. Okay, so this beta-lactamases enzyme is secreted by some microorganisms like E. coli and that Staphylococcus aureus. So actually, we see the structure of the penicillin and cephalosporin. They are the beta. They contains the beta-lactam ring here. So actually, this beta-lactam ring is closed in the form of closed. Okay. So this closed beta-lactam ring is very active on the and affects the antibiotic effect on that gram-positive bacteria cell wall. But due to the reason of some beta-lactamase enzyme action, what happening that the closed ring, the beta-lactam ring is due to the action of enzyme, they are opened. Okay, so this opened beta-lactam ring is inactive form. So they cannot able to act on that pathogen. Okay, so they become inactive state. From active state to inactive state by the reason of enzyme action, that is beta-lactamases enzyme. So beta-lactamase for penicillin and cephalosporin is called as penicillinases and cephalosporinases respectively. The another one is changes in the composition or structure of the target site. So these resistant genes also helping the bacteria to changes their structure composition, their chemical structure composition. It is also one more important antibiotic resistant reason. So they alternatively, the bacteria can add different chemical groups to the target structure. In this way, what happening, they are mostly shielding and protecting from the antibiotics. So how they expressing that alternative proteins means some bacteria are able to produce alternative proteins that can be used instead of the ones that are inhibited by the antibiotic. For example, it takes Staphylococcus aureus can acquire the resistance gene, that is MECA gene, and produce a new penicillin binding protein. Actual penicillin binding protein is altered in a new penicillin binding protein. So what happens when that antibiotic is targets at that region, they are not binds properly. So these proteins are needed for bacterial cell wall synthesis and are targets of beta-lactam antibiotics. So the new penicillin binding protein has low affinity to that beta-lactam antibiotics. So and is thus what happens that resistant to the drugs and the bacteria survive the treatment. So no effect more shown on that pathogen. So this type of resistance is seen in the methicillin resistant Staphylococcus aureus. Shortcutly we call it as MRSA. Okay. So till now I discussed some various types of drug resistance, how they are altering themselves, and some primary resistance, and the another one is innate resistant, and the another one is acquired resistant. How the plasmid is playing an important role to carrying the resistant genes from one bacteria to another genus bacteria. Okay, so now how we test these antibiotic sensitivity. So by testing the antibiotic sensitivity only, we can know the more their action, their concentration. So which concentration is suitable to inhibit so and so bacteria, like that. Understand? So these antibiotic sensitivity test is nothing but to determine the drug minimal inhibition concentration or minimal bactericidal concentration of the organism which is isolated from the patient sample. Understand? So in this process, we will take the folds of diluted concentration of the drug along with the sample isolated from the patient, and we placed it on a plate. Different concentration we placed on a plate. After the incubation period, we will see there the minimum inhibition concentration. Here 6.2 is the minimal inhibition concentration of that antibiotic sensitivity to that organism which is isolated from the patient. So the another way to study that antimicrobial sensitivity, antibiotic sensitivity, that is disk diffusion method. So in this disk diffusion method, we impregnated with various antibiotics strips we placed on the surface of the agar plates. So after the incubation period of 18 hours at the room temperature or at 35 degrees centigrade, the drug, what happens, the drug is diffuses outward from the disc wells, the disc, and the spreads the diameter. So the diameter of this zone of inhibition is determined and measured, and that zone of diameter is estimated the antibiotic sensitivity of that antibiotic sensitivity. So this, why we do this test means this provides the physician with a precise, with a specific concentration or particular concentration of a drug to guide the choice of the both drug and the dose. Understand? So coming to the conclusion. So usually the more often antibiotics are used, the bacteria adapt and find new ways to survive. So it is not a end one, it is a gradual one. So every moment they are evaluating themselves to enter into the host and to developing themselves to and altering, modifying their genes and undergoing the mutations and becoming very resistant to various antibiotics to continue to multiply and causing more harm. So all these because of the reason that is overuse and misuse of the antimicrobial medicines. Dear students, listen please. Overuse and misuse without professional guidance or without professional oversight of that by that professionals, if you use like that, what happens? The antimicrobial medicines are major factors that have contributed to the development of drug resistant of the microbes. So these antimicrobial resistance is a serious global public health issue that needs to be addressed immediately, and everyone has a role to play. Not you and me, everyone has a role to play. So to limits that overuse of the antibiotics and misuse of the antibiotics. So actually that thing only leading to that more drug resistant in the microbes. Understand? So the clinical pipeline of new antimicrobials is dry, as per WHO, saying that the clinical pipeline of new antimicrobials are dry. Means there is no there is more antibiotic shortages there in various countries in the health. For it is very necessary to identify and do research work on to identify and to develop a new antibacterial agents. It is necessary need in the healthcare systems as per World Health Organization. So furthermore, a lack of access to quality of antimicrobials remains also a major issue in the countries, in the healthcare systems. So my dear students, it's very needy of that innovative research or necessary to develop new antimicrobial microbials agents. I hope the becoming days will be good and evaluated many more evaluations to develop that drug that antibiotic to reduce this antibiotic resistance. Thank you one and all for listening me. Thank you.
Thank you so much, madam, for your wonderful presentation. It was comprehensive and it's a global need also. Recently in the Lancet journal, there has been an article on January 22nd about we need to rethink of funding for overcoming antimicrobial resistance, because the resistance that has been gained by microbes has pushed us into the pre-antibiotic era, where we had no antibiotics before penicillins. Overall, the talk, whatever you have given, it was comprehensive, and you have started from the basics, how the pathogenesis will be initiated, and what are the various mechanisms that are available for resistance of this, and what are the available drugs. And overall, it was wonderful. Thank you so much for giving a nice presentation, madam. Just as a token of our appreciation, we want to present the certificate to you. Just please look into this, madam. And this is the certificate. And thank you so much, madam. Thank you so much for your valuable time. Hope to associate with you in the near future also. Thank you so much.
Okay, thank you so much for giving me this opportunity. Thank you.
Thank you, madam. Have a nice day, madam. Bye-bye.
Okay, please share me the record and the certificate through mail.
Yes, madam. We will share.
Thank you. Thank you so much.
Thank you so much. Have a nice day. Bye.